Assalamualaikum and good day everyone. Today we from Group 6 would like to present on Practical 3, Identification of Enterobacteriaceae. Enterobacteriaceae are gram-negative, rod-shaped bacteria that are commonly found in the intestines of human and animals. They can also be found in soil, water, and food. This family includes important bacterial genera such as E. coli, Salmonella, Shigella, and Klebsiella. Some members of Enterobacteriaceae are harmless and normally live in the body, while others can cause disease such as infection and food poisoning. Because of this, accurate identification of these bacteria is very important in clinical diagnosis, environmental monitoring, and food safety. In this practical, different laboratory methods were used to differentiate the bacteria. These methods include the use of selective and differential media, as well as biochemical tests such as sugar fermentation and IMVIC tests. The objective of this practical were to isolate Enterobacteriaceae from samples, identify the bacteria based on their morphology and biochemical reactions, perform and interpret important biochemical tests, and understand their medical and environmental importance.
On day 1, the oxidase test is done for all cultures. The result is recorded if it is positive. And if it is negative, the experiment is continued by streaking the culture on McConkey agar. It is incubated at 37 degrees Celsius for 24 hours. On day 2, the result from McConkey agar is checked. The red colonies is inoculated for indole test, while white colonies is inoculated for H2S production. All media will be incubated at 37 degrees Celsius for 24 hours. On day 3, positive indole tests from red colonies are confirmed as E. coli, while positive H2S production will be further tested with indole test. This media will be incubated at 37 degrees Celsius for 24 hours. On day 4, the result from indole test from white colonies are checked.
Starting with our first plate, we utilize McConkey agar. This medium is selective for gram-negative rods. The isolate tested negative for oxidase. Our results showed distinct red colonies. This indicates the isolate is lactose fermentation, LF positive. The color changes occur because the pH indicator in the agar turns red under the acidic condition produced during fermentation. Moving to the eosin methylene blue (EMB) agar, we observe further characteristic of this gram-negative, oxidase-negative rod. The growth lack a metallic green sheen. The absence of this sheen suggests that E. coli is not present in this sample. Instead, this result suggests the isolate is likely another member of Enterobacteriaceae family. Finally, we conducted tube-based biochemical assays to narrow down the identification. For the right tube showed a negative indole result. The medium remained a straw yellow color after the addition of reagents. Based on this negative indole result, other possible identification for this specific isolate include Enterobacter aerogenes or Klebsiella pulmonea. H2S production. In the left tube, we observe a black precipitate, which signifies a positive result for hydrogen sulfide production. This happens when the bacteria reduce sodium thiosulfide into H2S gas, which then react with ferric ion to form the precipitate. This reaction is the characteristic of Citrobacter frundi. In conclusion, the result identified the presence of lactose-fermenting gram-negative enterobacteria within the sample, while the morphological and biochemical characteristics exclude E. coli. The significant H2S production and Indo-negative result indicate contamination by other coliforms or related genera common in the food safety analysis. This finding highlight the importance of using multiple selective and differential media to accurately distinguish between members of the Enterobacteriaceae family.
In conclusion, the practical successfully identified Enterobacteriaceae using various laboratory methods and biochemical tests. Although these bacteria share common characteristics such as being gram-negative and rod-shaped, they can be differentiated based on their biochemical reactions. Tests such as sugar fermentation and IMVIC were useful in identifying and distinguishing the bacteria accurately. Selective and differential media also played an important role by helping us observe differences in bacterial growth and colonial appearance. Through this practical, we gain a better understanding of bacterial isolation, identification techniques, and interpretation of biochemical test results. We also learn the importance of Enterobacteriaceae in medical, environmental, and food safety applications. Overall, this practical improved our laboratory skills and understanding our microbiological identification methods. That's all from us. Thank you.